Okay, so Section 8.3 deals with DNA replication, and we have talked about DNA replication when we were talking about mitosis and the cell cycle. And remember, we said the most important thing that happens during interphase happens during the S phase, and that's DNA replication. So in this section, we are going to talk about how that process happens a little bit more specifically. So DNA replication is the process that we make a copy of the genetic information for our cells that they can pass on to the next generation of cells, and this happens prior to cell division. So first of all, when you have replication occurring, that DNA strand, and remember our chromosomes are linear. They're just long strands of DNA, and we have 23 pairs of them. So 46 chromosomes or 46 strands of DNA that have to be replicated every time a cell is preparing to divide. So the nice thing, or the thing that makes DNA replication a little bit easier, is that our base pairing rules simplify the process. So we know that A always pairs with T, right? Adenine always pairs with thymine, and cytosine always pairs with guanine. And since that's the case, when you know one side of your DNA strand, you know the other side because of that complementary base pairing that we have. So during that S phase, the DNA replicates by splitting in half using what we call semi-conservative replication. Semi-conservative replication means that the DNA, the hydrogen bonds between our nucleotides or between our bases separate, and each side then is used as a template. And so we can use, or a pattern. And so you use those original strands and you add in new nucleotides to pair with the original. And so when you do that, that's going to create two new strands of DNA, and now all cells have a complete set of DNA. So how does this process occur? Well, it is a bit more involved, so we're going to kind of talk about the general process. DNA has to stay in the nucleus, right? And so it is the template for this process. It doesn't do anything other than get copied. What gets the copying done are other enzymes and other protein complexes that actually do the hard labor of replication. You have enzymes that unzip the double helix. That enzyme is referred to as helicase because it breaks the double helix, right? Helicase. And then you have nucleotides that are floating around in the nucleus. They're called free-floating nucleotides. They're there. They exist because of other molecules that are ingested by the organism and then digested and then sent on to the cells. So we have these free-floating nucleotides in the nucleus, and enzymes will bring in the right nucleotides. Those nucleotides will hydrogen bond and to the original template strand, forming two new strands. So besides helicase, right, and helicase is the enzyme that works to break these hydrogen bonds here or the ones on this side. But the other major enzyme involved is DNA polymerase. DNA polymerase is the name of it because it is the enzyme, it ends in ase, that is making the new DNA polymer, right? We know that a polymer is a big molecule. So this enzyme is creating that new big molecule. So DNA polymerase's job is to help those free nucleotides bond to the one before it, right, forming that new backbone. So it helps bond the phosphate of one nucleotide to the sugar of the other, creating those covalent bonds, forming our double helix. And so again, those are the polymerase enzymes help form covalent bonds. Hydrogen bonds just form on their own. So at the end of replication, you're going to have two new strands made, right? Because they have, the strand was opened up by helicase and the new nucleotides were brought in and paired up. And so in our picture here, we're representing that we have these two new strands. The original strand was the blue, right? So originally that DNA molecule was coded as blue, but during replication it opened up and the new strand here which is orange in color, paired up to the original strand. So now I have two new strands, but notice each new strand is composed of an original and a new strand. That's again why we call DNA replication semi-conservative, because we somewhat conserved the original molecule, right? Half of it stayed intact. Now the process of replication is very fast and it's also very accurate. For all the information that has to be replicated, it's amazing really how very few mistakes are actually made, pairing mistakes that are made. But if you look at this image, right, A is showing us these little bubbles here are where replication starts. So in eukaryotes, we have, right, we're eukaryotic. So in our chromosomes, there are multiple, what we call points of origin, right? These points of origin are where replication is going to start. And so helicase is going to travel in both directions from the origin out, right? So this one is going to replicate in both directions. This origin is going to replicate in both directions, and so does this one. And so you can see the replication bubbles. That's what we call these, areas on the molecule that have been separated. We call them replication bubbles. You can see them getting larger as helicase continues to unzip those hydrogen bonds. And eventually, right, they're replicating that DNA strand, and eventually those bubbles fuse, and you're going to get two new strands of DNA formed. Polymerase, besides adding nucleotides to the chain, there's actually several types of DNA polymerase. There's DNA polymerase 1 and 2 and 3. But a lot of them, almost all of them, can proofread and correct errors as they are found. So that's what cuts down on the mistakes, or what we would eventually call mutations, that happen during DNA replication, because DNA polymerase finds and corrects errors.